now I'm going to start the second part of this talk. Actually, the second part are more, we have more exercise to do, okay? The first one is the following. Remember I talked at the beginning, we want to be able to see the trajectory ends, to plot and record the data which is being created during the simulation. Okay, this is our task now, to plot the nucleus trajectory, because usually in the experiments, the nucleus is the best part to follow during an experiment. It can be easily colored by some dye, and with some fluorescence microscope, you can follow during its movement. Okay? So let's follow the nucleus and record the data. So now we can to not change the simulation you just created. We uploaded an extra simulation in the students folder, which is called something cell MIG 2D plot and rec, something like that. This is the one we are going to use right now. But I'm going to show you here how to change the previous one in the second one. So you have your previous simulation open in the Twilio here, and go directly to the steppables file. Locate your mouse in some empty part, and go to the CC3D Python tab, scientific plots, setup. This will create a plot window in your simulation like this. Set plot window, blah, 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 blah. It comes with some default lines here, with title in the axis, title in the window, and which plots I've been included in the graphing. Here, for example, we have the volume and the surface of a cell if the cell is in 3D. But we need to edit this one. We want to plot the nucleus center of mass. We want to follow the center of mass. So we have to change these lines to say that to the code. And this is done. Editing the title, nucleus CM trajectory, x-axis, put the x position of the center of mass, and the y-axis, the YCM, the position of the center of mass. And call this window, give it a name, Nucleus CM. And we are going to plot it with lines, color red, size 5. I can copy these lines for you and put in the chat window again. Okay, there you have it. So we have the plot window now. The next part of the thing is to say what to plot. Well, it means that we need to include in the start function vector, an array, to store the position of the center of the center of mass of the nucleus. This is done this way here. It's in blue here. See, the first line, I create a 3D array to store the current nucleus center of mass. The second array stores the previous position of the nucleus center of mass. In the third array, I store how much the nucleus has moved from one Monte Carlo step to another. Okay, I have to do that for one reason that I want to explain later. Let me copy these lines again. And put in the control copy. Here you have three arrays to store positions. The last line here, the loop over the cell type CITO. No, not CITO. I think I wrote the wrong name there. It should be Nucleus, but anyway, it was going to work here anyway. It's the where I store the initial position of the center of mass of the cell here. Okay, it could be nucleus here. It doesn't matter. So we have the start function. Everything is 0, but I save the old position as the current position of the center of mass, where the cell was created, actually. Okay. Now I'm going to step function. In the step function, I have to update this nucleus center of mass position. So I again, each time here that enters in the step function, he enters in this loop here, where I have I use the center of mass plugin here, XCon, YCon, take from the plugin center of mass the position of the cell type nucleus and calculate the difference between the current position and the old position here. Oops. The old position here. This is this displacement of the center of mass from the previous Monte Carlo step and this one. Okay. Then I save it in the old position to be used in the next time that the steppable is called, the actual position of the center of mass. Did you follow? This thing? I mean, I make the displacement between now and before.
And in step, okay, in the function step inside steppables class, you need to update your current position of nucleus based on the size of the step you made. Okay, you could plot your position directly, but there's a problem. Since we have a lattice with periodic boundary conditions, when the cell traverses one side, it will appear in the other side. So you will have your graph will be cut. Can you visualize it? The cell will stop at a point and show up at another point. So if you just plot directly the position of the cell, this will happen. That's why we update the current position with the step we made in the previous step. Okay, the displacement of the nucleus in the previous step. Are you with me here? Let's see the chat. Unfortunately, I cannot see the chat and present at the same time. I don't. Oh no, I can. I can. I'm seeing here. Okay. Did you follow until here? Is your code exactly as Gilberto's code? Okay. Do you need me to copy and paste some part of the code for you? Please. Okay, so I will copy and paste from line 48 to line 72 of the slide I'm showing right now. Okay. So I don't have his code loaded yet. Here. And steppables. It doesn't show the line. I'll have to download the simulation as well. Is it plotting the trajectory with PBC correction? What is PBC? I'm not sure.
Periodic boundary conditions, which is exactly what you're trying to do here.
Yeah, no, when you have the periodic boundary condition and you plot your point, it doesn't correct by the teleportation thing, you know. Your cell disappear here and appear there, but the plot doesn't correct. We have to do this by hand. That's why this code is a bit larger than you thought. So please understand that I was not aware. I was going to substitute Gilberto now, so. And I said from 48 to 72. What's happening? I cannot copy and paste. Maybe I have to close the windows first. I don't know why.
Gilberto, do you want to continue or do you want Pedro to continue?
I cannot copy and paste.
Yes.
Cecily, can you download the simulation from?
Gilberto is back, I think.
Gilberto is
Pedro,
back?
you want
Okay.
me to take over? My apologies, guy. My internet connection here failed for minutes and I was disconnected from everything. But now I can continue from.
I just explained why we are plotting the nucleus coordinates by calculating the displacement and not directly, because the CompuCell does not correct the fact that we have periodic boundary conditions and we need to save the displacement and correct it. Okay, so they already know that.
Okay, I'll share my screen here. Sorry, share screen 2. Let me go there. So you were here.
I think you're not sharing your screen or yet.
No?
No, I'm not seeing your screen at least.
No, it's okay. I think it's there.
Oh,
Gilberto, you were okay. You were sharing.
okay. So let me see, where did
There
you
we are.
stop?
Next,
Here. This part.
yes,
This is-
yes, this slide.
Okay, so as Pedro must told you, this part only takes into account of the fact that we are moving in an infinite plane, but we see only a part of it. So when the cell moves outside of the grid, we have to put it back in the other side of the grid. So if this displacer is bigger than certain amount, I have to discount that amount or increase that amount to the displacement to put the cell back in the window that you can see inside the simulation. Okay, this does everybody have part of it already? The code, the code. Did you cut and paste here in the chat window, Pedro? This part?
Yes, just did it. People are
Okay.
using right now.
So the last line is here. You update the current displacement, summing the displacement that you have calculated and corrected in the lines above. After you do that, you can add the displacement and that plot window, the graph window we have created in the start function. So you go to the CC3D Python scientific plots, add data points here, and add these lines to plot the actual position of the nucleus center of mass. So here I will copy these lines again. Control copy, Control V is that. And then after you do that, include this line, you are ready to go again. Save it. Every modifications you have done. Click on the CC3D logo, and the player will open with the graphical window aside of this main window, the cell filter. Sure. Put it to run, and you have something like that. Sure. Let me put it to run here. Open CCD. Since my network now is working. Here. File, open simulation, recording, plotting, CC3D. Right. See? Then you have the position of the nucleus being plotted in the graph trajectory window. Let's wait a little bit to your simulation works over there. Okay, so this is the end of this. Oh, no. There is an extra part of this, this lecture, this tutorial. That is now we need, as I told you before, also we have to export this data to be able to work with the data later. So how do you do that? You right-click on the plot window, and these options here will open. A drop-down menu will open here with the export option here. You click in this export option, and you have here the second window being open. And it asks you the export format. You want your data that is plotted in this window be saved. So you choose whatever you want here. Matplotlib, blah, 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 image, or however. You can save this window as a window, GIF, PNG or TIFF. But we wanted CSV, comma separated value from plot data. It's an Excel file like that. So you choose this one, choose what the separator you want for the data, comma. You can choose another option here, for example, tab separator between data, and click export. As soon as you do that, a file will be saved in your project folder here. Okay, and this actually, sorry, this window will be open to naming the file you want to save. For example, I am saving here as with the name nuctrash. It's a CSV file. Then save it. The saved file has this content: 2 columns, X center of mass, Y center of mass. And now you can do whatever treatment of data you want with those data, to calculate the mean displacement, the mean square displaced, to calculate the speed, the velocity autocorrelation function from this data here, because you know the time interval, time in terms of Monte Carlo step that this data is being saved. It saves it at each Monte Carlo steps, and the time interval is 1, for example. Okay, and that's it. You have the data saved and the window with your simulation to be observed during on demand, let's say, on the fly, on running. Okay, this ends this part of the talk. The 2 and 3 main goals that I proposed from the beginning. The next one, can I go forward? Let's see in the poll. This one is the last one. I'm going to. Okay, 72% is doing the exercise. Okay. The next exercise will be presented by Pedro. This is a very nice simulation he did. And in this simulation, he will extend the previous one, the first one, the single cell simulation one, to include the multiple cells. Okay? Now, Pedro, take over. Thank
Okay,
you, guys.
thanks.
I will stop sharing here.
Yes, okay. I will activate my camera. Just so you guys know my face. Let's here, start video. And there we go. Perfect. So I am a student from of Gilberto. Gilberto, I am a graduate student, and we work with cell crawling. We work with isotropic conditions, which means no chemotaxis, no fields, and with anisotropic conditions as well. So maybe we can show you later if you have time. But for now, I'm going to teach you how to build this simulation with multi-cell. So using this simulation, how can we introduce more cells? Okay. First question, what happens if we only create more cells with no caution? What do you think would happen if we only create more cells? Let's see what you think. Am I screen sharing?
You're not screen sharing yet. No.
Okay. Now I am.
Yes.
Perfect.
Okay. They will merge. Nice. You are too good, actually. So this is what will happen. They will bump into each other, yes, but they would bump anyway. So this is a cut from a simulation, and the nucleus are merging, the cytoplasm are merging. So this doesn't make any sense. This video doesn't seem right for you, right? Myofiber, yes, could be a myofiber, yes. But the nuclei wouldn't merge, right? So this way, I think you couldn't construct even a myofiber because the nuclei are separated. No, they do not. So this is a wrong simulation. Okay. Okay, Suvik. What do we want to happen? We want this. We want the cells to crawl individually. Yes, they will bump into each other, but they won't merge. Okay, this is what we want. But they are crawling individually. Yes, we can change that. We can change that with contact energies, but simply adding more cells, we won't reach the simulation we want. Okay. So first of all, how to achieve this? We need to build our cells in a way that the compartments that constitute the cell belongs to something that is different from the other cell, which is built with the same compartments. Multinucleated giant cells in TB. I don't know that. I don't know about that, but if you can share, later will be fine. And more than that, we need that those compartments inside the cell behave differently than compartments from different cell, you know? So for example, here we have a set of cells that I'm going to refer as a cluster now. So we have the cluster 1 composed by compartments 1, 2, and 3. And we have the cluster 2 composed by the compartments 4, 5, and 6. So what do we want? We want that the compartment 4 to have a to have an interaction with the cell, with the third cell, different from the interaction it has with the fifth cell, even if the third cell and the fifth cell are of the same type. You know, so we need to some way make those compartments belong to a cluster, to something new. And after we've done that, we need to give different energies for compartments that are interacting inside the cluster and compartments that are interacting outside the cluster. So there are two types of energies we are going to use now. We are going to use the contact energy, which you already know, and we are going to use the contact internal energy as well. So the contact internal are the energies of the compartments that are interacting inside the cell, and the contact will be the energies that will mediate the contact of compartments from different cells. Okay, so the Hamiltonian will be a bit different. It will use the J internal for same cluster, for compartments of the same cluster, and it will use the J external when the clusters are different. Okay, I bet it's clear for you. So first of all, let's start with the most basic simulation you have, which is the CellMig 2D. Open the CellMig 2D folder, which you can download from the drive, and load the CC3D project onto edit. So this should be your simulation, CellMig 2D. Open it and load the CC3D file onto edit. Once you've done that, your simulation should be just like mine here on edit, and stay with the XML file opened. Let's make it a poll. You just need to load the CC3D project inside the CellMig 2D file folder. Okay, you're good. In the XML, delete the uniform initializer steppable. So go to the end of the steppable, okay, here, and delete the XML, and delete all the steppable module here. It's called uniform initializer. We won't use it because we want to draw the cells by hand, okay? We will draw the cells using Python after. So we are not using it right now. In Python, you can draw the cell in the shape you like. It's absolute free. So it's also interesting that we teach you how to do this at some time, right? So delete everything. No, not everything, just the uniform initializer. And set the chemotexes lambda to -150. So you go to the chat is. So you go to here. It's line 60. And you change the lambda to minus 150. It will turn it will make this force stronger, okay? Because the lambda regulates how strong the lamella will invade the medium. So when you increase in modulus, when you change it to further negative values, you make this force stronger, okay? You increase the probability that a lamella pixel will copy over a medium pixel. And after you've done that, I want you to start a new contact plugin, which is called the contact internal, but don't do it still, okay? Don't do it yet. I will first explain how it's done, and then I will copy and paste the code to you. So it will be faster this way. So what we want to do? What we want to do? We want to introduce the contact internal plugin and also the contact plugin, and set the contact internal energies to be different from that from the contact plugin, so we can control how they interact when the compartments belong to different clusters. So your code will look like this. You have the contact plugin with some energies and the contact internal plugin. In the contact internal, you are basically saying how the compartments will behave inside the cluster, and outside the cluster on the plugin contact. I am gonna copy and paste it for you. But I think you can do it you could do it by your own if you needed. Because it's an option available in the. I cannot copy and paste large things, so I will have to copy and paste by pieces. So this is the first piece, the contact. Notice that the energies are different. So those energies are different because we adjusted them. And the other piece of code is the contact internal, which I also sent to you. So your code should look like my code, which is in the right side of the screen. It's a really difficult job to adjust those energies. Did you pass the code? Yes, I passed it directly to Giuliano. I'm sorry, guys. I passed it to. I always committed a mistake. Always. So here's the first one. And here's the second one. Sorry, guys. Okay. Are you good? I'm going to continue now. What did you change before contact internal? Before the contact internal change, I deleted the uniform initializer steppable on the end of the XML file because we are going to draw the cells using Python. So at first I did this, And I also need to do this in my simulation here. I'm sorry, forgot that. Here. So it should look like this. And save. How did you select these contact energies? I tested very hard many types, many combinations. It's basically a parameter scan that I did until I got the format I wanted, and I mean the shape of the cell I wanted, and the speed I wanted to look, just visual, you know. But it's hard work. If you wanted to do this, it would be from scratch. It would be hard work. Contact internal is for energy between compartments of a single cell. Yes, which I am calling cluster. This cell, biologically speaking, we are calling cluster in CompuCell terms. And contact energy is for between different cells, which would be different clusters. Yes, that's it. So now I will proceed. And here. So now we need to create the compartments. Okay. What did we do until now? We set the energies, just the energies, and deleted the cell creation. Now we need to create the cell. So go to steppables. I mean cell mig 2D steppables.py, and go to your start function and give a new line there. And we are adding a piece of code to create each cell. We need 6 cells. So I am building 6 cells. And each of those cells must have a lamel, a cyto, I mean lamellipodium, cyto, and nucleus. Okay. So this piece of code is doing exactly that. And then I need to assign a set of lamel, cyto, and nucleus to a cluster. Okay, these are the 2 things I'm doing here. So look at the code now. I'm gonna pass and copy and paste after, but pay attention here. So first of all, I am setting our number of cells to 6. This is the maximum here in the way the simulation was done. The 6 cells is the maximum. And I am also defining some lists and a matrix of X and Y coordinates of all our cells. 10 and 50 are the coordinates of the first cell. The first cell to be created will have this set of coordinates here. The other cells will be created. They will be created on the side of the previous one. So we introduce the first coordinate, and then we change it. We give it an append to incorporate the other coordinates of the other cells. So to create all the cells with all the compartments, we do this loop over our n cells, and we use the command pots create cell to create a cell, a new cell. We don't know yet what it would be, but then we say the type of the cell will be cyto. So we are creating a cell of the cyto type. Okay, and then we give an append. We give an append of this cell in the list of the CIT, in the CIT list, which is the list of the cyto compartments. Okay. Then we do the same thing for lamellipodium and for nucleus. So create, assign a type, and put it into a list. After that, we need to tell that this lamellipodium and this nucleus belong to the same cluster as that cyto here. So we will use this function, reassign cluster ID, and take the number, the ID, the cluster ID of the cell cyt, which is the cyto cell, and pass it into the cell lam, which is the lamellipodium cell. And we do this We do the same thing for the cell nucleus. And then we append a new set of coordinates inside our XY CM matrix. Okay, we have a chat. Let's see. Yes, exactly what James said. So I'm gonna copy and paste. I will take it from my simulation here. So it's all this piece of code here. I'll try to, yes, I can paste on the chat. And I'm gonna paste as well on my simulation here. So I am doing with you. So if some bug happens to me, it will probably happen to you as well, and we will be synchronized. So copy and paste. Did you understand? Code not here again. I'm sending only to Ajay. I forgot this. I always forget this to send to everyone. Okay, so this is the code. Copy and paste on the start function. Okay, so it should look like exactly as my simulation here on the right. Obviously not, yes, exactly like my simulation. And then you need to start a poll. Relaunch. There we go. So if you don't understand some part of the code, please ask on the chat, okay? Because you are not programming directly, so make sure you understand at least. And if you need help, please comment on the chat as well. Cells are not appearing. Yes, not yet. Explain line 30. Line 30, okay. So I have a matrix of X and Y coordinates of my cell, of all my cells, okay? So I have 6 cells. How will my matrix look like? I'm sorry, I'm using paint, but you know, whatever. I have 6 cells. So here's the first cell, the second one, the third one, the fourth one, the 6th one, the 5th one, etc. And here I will have a sublist of X and Y coordinates of the first cell, X and Y coordinates of the second cell, etc. Okay? So now look at my code. I introduced the first set of coordinates for the first cell. When I pass through this loop once, I have to append to this matrix the coordinates of the next cell and so on. So did you understand that? So to calculate the next position, I just do a plus 20 increment here on the X coordinate. So I just so I will create this other cell 20 pixels on the right of the previous cell. Okay, this is exactly what this line is doing. Did you understand? Got it. Nice. Okay, let's get back to the. So I think you all done that. Now we are going to create the cell effectively. I mean, we already created in the CompuCell software, but we didn't draw it. We didn't paint the cell in the lattice per se. And we didn't do it using the initializer steppable as well. So we are going to do this programming with Python. So the same thing, I'm gonna copy and paste the code for you, but please pay attention and understand what I'm doing here. first, I need my initial condition to be just like this. It's here I'm showing 4, but it will be 6 cells, okay? And each cell, each of these giant rectangles there are composed by 3 square. The lowest square is the cyto, on the middle is the nucleus, and on the top is the lamel, okay? Lamellipodium. So I want to create it this way. To do this, I have this other piece of code here, okay? Same thing. Stay with me, try to understand, and then I copy and paste you. So first of all, I am introducing an object and a point object from CompuCell, which basically represents a pixel in our lattice, okay? And we need to create all our n cells. So first of all, I'm doing a loop here over all n cells. And then inside this loop, other 2 loops over x coordinate and y coordinates, okay, from -3 to 3. So basically this is a 6 per 6 square, no, 6 by 6 pixels. And we assign the x and y coordinates to our point PT. And we use this command cellfield.set to say, in this point PT, we want to have a cell of the cytotype and of the cluster ID NC. And the same thing for the nucleus and for the lamel. Okay, so here I am assigning this point PT to the nucleus, to a nucleus that belong to the cluster ID NC. Same thing for lamel. The only difference between the 3 is that I am increasing the y coordinate, so I create one above the other. Is it clear? If it's not clear, please please tell me on the chat. Now what I'm going to do is I'm going to copy. Where is it? It's not here, it's here. So this is our piece of code that I'm sending to you now. And I'm also pasting it in my simulation. No, again. What again? Again, no. What again? Other piece of code? Again. Yes. I think it automatically set to send to someone because I was sending to everyone before. I think it's some bug. Where is the NC list where the values are getting updated? Right, good question. So look here at my code. So the NC starts with this 4 here, okay? When those 3 cells are being created, they are created with the same NC index, okay? So whenever I make another loop over NC, I will always be getting those 3 cells of the same cluster, because from the beginning they were assigned to the cluster ID NC. I'm getting a strange error for the all codes in module 3. Unrecognized arguments. Can someone help him? I don't know if I can. I shouldn't stop to solve this problem. So someone please try helping Suvik. Okay. Now I'm gonna make a poll here. Okay. your code should look just like mine right now. Okay, so only 2 people need more time. All the rest are done, so it's done, I mean. So I'm gonna move forward. Okay, so we draw the cells now. The last thing we need to do, and this is the last thing, we need to set other volumes for our cells, and we need to do this because we need smaller cells so they can fit in a more beautiful way in the lattice. And we also, and I am also decreasing the size of the nucleus, so it decreases the chance that the nucleus would break up the cell. So this is a way I found to solve that issue. So first of all, you can reduce the size of the nucleus so the cell don't break up, because with a large nucleus it has much more inertia. Obviously, you can't talk about inertia in pots, but I will leave it as this. And also fixing those energies, you can also do this. Okay. So by now you can run just like Forest. So where is here? Let me open my CC3D and show you how it goes. It's not opening. Ah, it is. No. Right here. So I click a step. Okay, some people don't know about this. I click a step first. I don't hit run. Okay, I don't give a play. I click a step so I can see the initial condition. Okay, I always do this. Okay, this is my simulation. And I also give 2 or 3 more steps just to make sure, and then give it a play. And I am unlucky, one of the nucleus disappear. Let me run again. And it's working, at least for me. Let's see the chat. Tá. Beleza. Could you do this? Mine looks the exact same as yours, except one lamel completely separates. Mine are stuck together. Yes, so probably your energies are not the same as mine. Okay, for all 6. Yes. If it's for all 6 cells, probably your energies are different from mine. So let me get back to my energies and show you the values here. Probably your values are different. Please check it out. Mine are all breaking. Check the energies. Check if contact internal, if it's contact internal here and contact over here. Check the neighbor order as well. Must be 4 in both of them. Okay. Must be neighbor order 4. So C to 15, C to lamel 22, and it's different. It's exactly the same of mine. I didn't set my lambda to minus 150. Yeah, this shouldn't be a problem. Did you check if it's contact internal and contact? Because all I did is exactly what I said. So I made it live so you could see. It was the lambda? Oh, okay, okay. Nice, nice. So Paulo had the issue and also Joseph. Mine was minus 500. Yes. What happens when it's minus 500? The lamellipodium will try to invade the medium as much as it can, right? So it will disattach from the C2 completely. I mispaced it, okay? Missing the second lamel and nucleus, okay. Nucleus energy statements. Okay, so I can copy and paste again. No worries. And now I'm sending to everyone this and this. With this presentation, we are able to perform cell division in the same way as, yes, you can. Everything is allowed, even with contact internal. But, many buts, you need to make sure your created cells belong to the same cluster. So every time you create a new cell, you must use that command cluster reassign cluster ID. Okay? You must do this every time you create a new cell. If you want that cell to belong to a cluster, you need to say it. So with this, guys, you can make something you couldn't do before, which is, for example, let's say you have two groups of 10 people. One group love people love between them, the other same thing, but the two groups hate each other, and they segregate. They are all of them are people, right? So they are like cells of the same type, but they hate each other. They love each other in the same group, but they hate each other. This very, very simple model, you need to, if you want the cells to be of the same type, right? You need to tell me the error, Tahnim. You need to use contact internal and contact. Now I understood your question. Sorry. If you want to perform a cell division, yes, you would have to divide all three compartments, or you could divide only the nucleus and make the nucleus create the other cells. It's also another option. And Juliana said there is a cell division for clusters. Perfect. Okay. Ajay and Tahrim, if you want to go to a breakout room. Okay, so I will proceed with the presentation now. I hope everyone at least understood what was done. Okay. So now you have an exercise to do. You have a homework. The homework is to create a steering panel. So a steering panel is something very key here, okay? Because adjusting those energies is very, very hard work. You need to, you go to your file and you change here, here, and there, and then you run the simulation, then you go back, and then you change here, 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 and there. And this is very difficult, mainly because all those energies, they they form like a system, a multivariable system, where you change one energy and this effect will change another effect in your simulation. So it's like trying to tuning a bad guitar, a really bad guitar. You probably know what I mean. You tune this chord here and this string here, and the other string get out of tune, and vice versa, you know. So it's very difficult. With the steering panel, it will be much more easy, because you will have those buttons you can adjust, and then with the simulation running, you don't even need to pause the simulation. You can adjust your energies during the simulation, and also you can adjust the Lamel target volume, the Lamel lambda volume. You can adjust everything. Okay, you can adjust volumes, energies, lambdas, and also the chemical lambda. Okay, so this is your homework. We have a demo for that. So if you installed CC3D in your PC, you have a demo called steering panel. So you can base yourself in this demo to make this homework. And this is very useful for you. We also have the result. So the simulation result is also uploaded on the drive. So if you want to jump straight ahead to the simulation, see how it's done, it's there. Ah, right. So I'm actually gonna show you this code now. Let me just pause the simulation and open the simulation with the steering panel. So where is it? I think it's downloads. Yes, steering here. So use the single cell simulation, please. You don't want to suffer extra. So this is the steering panel. Okay, are you with me? So the steering panel is an amazing piece of work, where you just change these guys here from up to down values here, and while the simulation is running, you'll see the effect. So I'll hit play here, and I will increase lambda volume, and boom, look at the lamellipodium now. So now you have a completely different cell, right? So the shape changed, everything changed, the speed of the cell changed. So I can reduce the C2, for example. Let me reduce much more. I can make an egg. So if I do this, for example, and let's make this. So probably I will make an egg doing this, not quite. So let's change the chemical lambda again and see what happens. So the cell broke up, but it catches right away. So broke up again. And you can play a lot with it. So you can vary your parameters using the steering panel. It's much more easier. Let's try other parameters. And I mean, you can put all the energies here on the steering panel, right? So look, bigger C2. You can make a extremely elongated cell, but it breaks up. you can avoid the cell breaking up if you add some more how can I say some more plugins, like that plugin that how it's called, let me see, plugins I don't remember, it's not here, but you can add a plugin that avoids cells from breaking up. You can do a lot of things and put everything on the steering panel. Just one more thing, be careful because steering panel consumes processing time. Okay, so if you are trying to do a really fast simulation, you probably will want to vary your parameters outside the yes, of course, I will almost forgot. Let's open here. Okay, and here. Ooh, I closed the window in the left side here. Let's go to the notepad. Okay, so here I have the simulation with the steering panel, and what you want to add here, it's the following. First, every class you create on Steppable should be called on your file .py, your .py file, not Steppable. You know, that third file you don't hear much about. So you have the XML, the Steppables py, and the py. Okay. So in the py, you always call the classes you are introducing. For example, here in steering panel, I am introducing here a class steering steppable. Okay, so this class is being called here on the py file. So you will have register steppable, steering steppable here, and also calling in the import head. Okay, so make sure you always call your class. In the XML, if you want, for example, to put in your steering panel the volumes, you have to delete the volumes here in the plugin on XML, because if you set some volume in the XML, it will overwrite the volume you are setting on the parameter scan. This volume is higher on the hierarchy of the values, you know. So if you want to add a value if you want to change a value in steering panel, you need to delete from your volumes here from the XML. Okay. If you want to change an energy, so it's not done here, but I can show you how. You need to, I don't remember the syntax exactly, but you need to introduce an ID for a specific energy. So if I want to change medium nucleus energy, I need to make an ID, for example, like ID equals nuc Maj energy, something like that. And in the simulation, I can access this energy by calling it nuc med energy. Let's see if I find it in some simulation here. No. Instantiate the steering panel into yes. Oh, I cannot use my tweeted because I closed the left window. How can I? No. I'll try resetting the. CC3D project show project panel. Okay. Or resetting Twitch. Okay. Here. So if I wanted to do it from scratch, let's say, I would be here in Steppables. And let us open a clear space here. And I would go here. So CC3D project. No, CC3D Python. And you go to steering panel and set up a steering panel. Where are the classes? It should create the class as well.
Hey, Pedro, this is potentially a pretty detailed bit of technical work for how to do this. How do you feel about can grab the link in the documentation for how to do this for now, for those who might be interested, and then we can go through it a bit more rigorously in a later session this week.
Yes, I agree.
Great. Okay, I'll grab that link. Yeah, so you don't have to worry about it.
Yeah. So there's a demo on the demos inside the installation folder of CompuCell that you can check out. We also have uploaded the simulation with steering panel. And also check out the documentation Giuliano posted on the chat, which is the manual. Okay, so thanks for your patience.
Okay, thank you very much. I mean, these are sophisticated things, and so it's fine to, if you didn't follow all of that, don't worry, you'll get to see it again. I appreciate Pedro being willing to step in for Gilberto when there was a technical problem. So are there other questions about this particular module before we close up for the day? There's a question.
Yeah, I have a quick question. This might be technical. So, so I looked at the cheat sheet, but I didn't saw is that the opposite. So those the properties for self is not is it built-in attributes?
Which attributes are you talking about?
I'm talking about like the cell mic to the stepable dot pi. So if you look at the self dot POSN.
Cell dot.
Yeah. Like DS1N and OPOSN. Yeah.
Okay, here.
Yeah.
Here
Are
you mean?
these?
Okay.
Yes. Are these attribute are built-in? Because I didn't find it in the cheat sheet for the attributes.
So this here is Python programmation. It's not it has nothing to do with the CompCell itself. So this is generic. You are creating a variable that belongs to the entire class. So when it has the self dot before, it means you can use it between among all the functions inside the same class. So I could create another cell like self Pedro here and tell it is nice. And then I take this variable here and I jump to another function and I can print this variable with no problem. Okay, so those things here are just created things here in the simulation that we use to pass from one function to the other. So in this case, why are we using the self dot some name, right, some variable in this simulation here? It's because we need to define it. We need to define them in the beginning of the simulation, in the start. So, and then use them after in the step. So in order to do this, we define them as self variables.
Okay, thank you.
Multinucleated cells, like giant multinucleated. How would we? Probably we would have to create like different nucleus types. And if you want a variable number of nucleus, which is very real, right? So for example, muscle cells have a variety of numbers of nuclei. So if you want to do this, probably you would want to I don't know how to do it actually. Not right now, but it can be extended to multinucleated. Yes, you can. If you want a variable number of nuclei, it's gonna be much more difficult, I think.
No, I think it'll work. You can create compartments, if the nuclei don't adhere to each other,
Yes.
you can create as many as you want of the same subcellular type within a cell. It'll work fine.
Yes. Yeah.
In
Yes.
fact, the simulation you have will do it if you like.
Let's see if there's another question here.
I'm sorry, if you use the contact energy at the same time adhesion flex, I mean by introducing some cations and these kind of things in order to define adhesion energy. So which one would be in the effect? I mean, do they work at the same time, these two plugins?
I didn't try this, so I cannot answer the question. Sorry, I didn't try. I don't know what would happen. Maybe James.
Sorry, could you say that again, please? I didn't catch that.
Is it possible to use the contact plugin at the same time with adhesion flex plugin in order to introduce contact energies?
You probably would want to just use adhesion flex. You don't really need both. And I don't know, TJ, if they can run together.
They can, and it would have an additive effect, but
That's what I expected.
yeah, but it would be a major redundancy and also pretty unnecessarily expensive computationally, because anything, as James said, anything you could do with the contact plugin, you could do in adhesion flex.
Wow, yeah, thank you.